So I'm going to tell you today about the power of miniaturization in medicine, in particular as it relates to cancer. We've been hearing a lot about today the Moore's Law. Mostly we've been hearing about it as the thing that's been disrupted by the price of DNA sequencing. I actually want to just say that Moore's Law has done well by all of us. We can now fit 1 billion transistors where we used to fit 1. And that made computation portable, and that's changed all of our lives. And so in my lab at MIT, we're very interested in what this miniaturization revolution can actually do for cancer detection and therapy. So at this point in the cancer session, you've already heard that there's an extraordinary opportunity to save lives by early cancer detection and prevention. Worldwide, about 2/3 of cancer deaths today are probably preventable by techniques we already have in hand. And we know by 2020, 70% of these will be in the developing world. So the way we think about detecting cancer and actioning it really fundamentally need to change to meet that challenge. So what can miniaturization do to help? This is an image from a colleague of mine at Rice University on the right. Her name is Rebecca Richards-Cordham. She actually just won the MacArthur for this work, so she's a fabulous colleague. And what I'm showing here is, on the left, a standard microscope, and you've already heard about the pathology microscope and how this might be changing. But this is today how we look at a biopsy to do cancer detection and diagnosis. So one thing you can do for miniaturization is miniaturize that whole microscope into this part on the right. This is a fiber optic microscope that Rebecca and her colleagues made in the lab, and it fits on the end of an optical fiber. And this changes the paradigm, because instead of taking a sample from the patient and looking at it under the microscope with a pathologist, now you're bringing the microscope to the patient. Okay, so that's one form of disruption that miniaturization can achieve. Now, in engineering, we think of this as sort of straight-up miniaturization. You took a big thing and you made it little. But I was talking to you before about portable computation that came from aggressive miniaturization. So you might ask, how can we completely disrupt this space by further miniaturization? So what if we had a detector that was so small that it could circulate in the body, it could find the cancer all by itself, and it could send a signal to the outside world? That could be truly disruptive. And in our group and several others, we think that nanotechnology can get us there. So just to kind of orient you to length scale, a human hair is 100 microns. So these are particles that are 1,000 times smaller than that. And they're small enough to circulate. Let me show you how we think they could work. So this is a blood vessel in the body. We're injecting nanoparticles in the blood vessel, and we're watching how they travel from the blood into the tumor. So the blue nanoparticles here are 100 nanometers, and it turns out that many vessels are leaky, and these 100 nanometer particles can leak out of the blood vessel into the tumor. Now, in our group, what we've done is designed these 100 nanometer sensors to detect chemicals that tumors make as they're invading. So they're a special class of enzymes, and these enzymes are catalytic. So every copy of one enzyme can do 1,000 chemical reactions. And that means that these sensors are ultra-sensitive, okay? So the next thing we need to do is get this activated signal out of the body so that we can act on it as physicians. And here we use another trick of the body, which is that the kidney is actually a nanoscale filter. The kidney is a 5 nanometer filter. So in this graphic, what you're seeing is if I give 100 nanometer material, it will recirculate in the bloodstream. But this 5 nanometer material will actually be concentrated and come out into the urine. Okay? So we can put all of this together as follows. I give a 100 nanometer particle, it can leak into the tumor. It can detect enzymes ultra-sensitively and get activated, and it gives off this tiny little 5 nanometer fragment, which the body concentrates for us in the urine. And now we have a urine test for cancer. Okay? So the way we see this playing out is that you can detect a signal in the urine that's of interest. And if you think about the developing world population, what might be particularly attractive is an inexpensive point-of-care test for this. We heard about this earlier. You can actually put tests like this on paper. right? These are analytes that are completely synthetic. We call these synthetic biomarkers, so we can design them to be compatible with readouts of choice. So the way we imagine this playing out is that you would get a shot, wait an hour, and do a urine test on a paper strip. And this would be a way to do a cancer diagnosis that could be done all over the world. That might not need steady electricity, might not need a physician on site. You could take an image of this device with a smartphone and send it off remotely. So that's one thing that we think that nanotechnology might have to offer. Now, I know that many of you are physicians, so just to add a little bit of molecular decoration around this, what are we actually detecting? This is a class of enzymes that we're detecting. They're called endoproteases, and they are responsible for tumor invasion. There are things like collagenases and gelatinases and stromolysins and MMPs and sort of many of your favorite enzymes, and they remodel all of the scaffolding of the human body. And as I said, there's over 500 of them in the genome. And if you just look at any cancer review article, you see that these are upregulated through all the phases of cancer growth, survival, invasion, and spread, including, importantly, the immune response and immuno-oncology. So we can make detectors against any of these. Okay? And this is important because if I just have an ultra-sensitive cancer test, that's actually a problem, right? Prostate-specific antigen, PSA, has led classically to lots of overdiagnosis, right? It's good for some things, but it's not a great screening test. And that's because if you crank up sensitivity, then you can sacrifice specificity. So to get both, you need to make multiple measurements. Okay, you need to do multiplexing. So how can I multiplex this technology? Well, I don't have to just make one sensor against one enzyme. I can make a cocktail of sensors against different enzymes and point them at different cancer targets or different phases of cancer. And in fact, that's what we do. I'm not going to explain all the details behind this, but we typically do a 10-plex. And what we do is we make it so that every fragment that comes out in the urine is designed against a different enzyme, and they're barcoded. Okay, so this is a 10 barcode. This is a mass spec barcode. You can barcode on paper. You can barcode by DNA sequencing. These materials are now completely synthetic, so whatever your tool is that you're interested in making a measurement with, you can design the biomarker so it goes with your tool. So let's just take an example of how this might play out. So let's look at lung cancer. So we know that there are many deaths worldwide, that this is growing, that there's now a recommendation, at least in the US, to do low-dose CT scanning of high-risk patients. Okay? So 24% of patients that are scanned because they're at high risk for lung cancer will have positive results. 95% of those are false positive results. Okay? And those patients need to be followed up, either a repeat CT or an invasive biopsy with bronchoscopy or a PET scan. Okay, so you might ask, in that setting, can I have a synthetic biomarker, a shot and a urine test that can stratify that patient? And furthermore, can I say, if this is the radiographic finding of this patient, is it malignant or not malignant? And if it's not malignant, what else might it be? Is it COPD? Is it interstitial lung disease? There are many other diseases of the lung that could give false positive radiographic findings, and since you have a multiplex, you may be able to stratify patients not only for malignancy or not, but in non-malignant cases, what else do they have? So it's a very exciting time, actually, because it turns out that in order to design a test like this, since this is a synthetic biomarker, we don't have to go out and just collect blood from lots of patients and do pattern recognition. We can take genomic data that already exists. So we have the Cancer Genome Atlas for lung cancer, and it turns out we also have a genome atlas for normal lungs and for the other diseases I described. And you just do a data mining exercise, and this is what my students did in the group, and these are the enzymes that come up. So of the 550, these are the top 20. These are the ones that you should make the sensors against. And if I go and I make a heat map, these three that are shown on the right can differentiate between cancer, COPD, and interstitial lung disease. So I just show you this as an example, but you can do this for any cancer for which you have genomic data. And in the lab, we've done this for colorectal cancer, ovarian cancer, and we're working on several others. So we design the sensors, then we ask, okay, does this work? We go into mouse models. This is a fancy genetically engineered mouse model that Tyler Jacks made for us in the lab. And this is cool because this is a synthetic biomarker approach, and so now I can give the sensor any way I want. I don't have to give it as a shot. I can give it as an inhalable nanoparticle. Okay, so now this is an inhaled nanoparticle. You wait an hour, you do a urine test on this mouse, and you monitor tumor progression in this genetic mouse model of lung cancer. And here you can see when we give the inhaled particles, they get into the lungs, they get around the tumor, and now you can see in the urine of this animal that by week 3, they have no detectable lung cancer on imaging, and they look perfectly healthy, that you can start to see the urine biomarkers coming up. And by week 5, we have a perfect area under the curve in mice for detecting early cancer through an inhaled test and a urine detection. So that's just one thing that you could do. What else could you do? Well, you can think about prevention. So here's another case. This is fatty liver disease. We've been talking about this today. This leads to cirrhosis, and it puts you at risk for hepatocellular carcinoma. You can look in these patients to see who is progressing, who is getting cirrhosis, who is making scar, and who should come on one of the new drugs that's being developed by pharma. There are 50 drugs in the pharma pipeline against NASH, this particular disease. So you can stratify these patients using, again, a shot and a urine test. You could stratify them for therapies. This is a new class of therapy that's come out of a Bay Area company called Cytomics. These are antibodies, and these antibodies are activated by enzymes. And so they have a class of these antibodies, for example, ones in the immuno-oncology space, and the idea is that they would circulate in your body and not be active. And only when they enter into this tumor microenvironment where the enzymes can activate the antibodies do they actually act, and that would prevent side effects. So here, you need to know what enzymes are active in the tumor to deliver this therapy. And so we can stratify these patients for these antibody classes, so they're companion diagnostics. And finally, if the urine test turns positive, then what are you going to do about it? So you could put them on drug, you might stratify them for therapy, but in the touching case that we just heard of, liver metastases, you may want to direct surgical intervention. And so here's a version of the probe where we can load it with a PET tracer. Okay, so the scenario is patients being screened by a very sensitive urine biomarker test. Urine biomarker turns positive. You load the same exact FDA-approved probe with an FDA-approved PET tracer, and now you can do very sensitive imaging in that setting and direct surgery. So we think that there's not just drug therapy that this can inform, but it can also inform interventional radiology. Okay, so I'm going to just pause here and say this is just one half of the equation, which is early detection and prevention and what one does when one detects cancer early. The second half, of course, is what can you do in nanotechnology with improved therapies. I want to pause and just say today is 10/9, October 9th. It's actually the first National Nanotechnology Day. It's named for 10 to the -9, which is the length scale of nanotechnology. If you go on nano.gov, you'll see that there's actually a lot going on in Washington right now. But separately, there turns out to be a whole set of National Nanotechnology Day initiatives that you might be interested in. So what might one do, not just in detection, but in therapy in the National Nanotechnology Initiative? So this is an idea that's been out there that you may have heard of, and the idea here is to make these same nanomaterials that can find their ways into tumors and not have them be sensors, but actually have them deliver chemotherapy locally. These are so-called targeted nanomaterials. And historically, this has been done with the classic chemotherapeutics, the cytotoxics, but this is now also being done with some of the newer agents, the molecularly targeted therapies. Okay, and what happens is you package these nanomaterials, you package these drugs into nanomaterials, you decorate them with stealth coatings that help avoid the filtration of the liver and the spleen, although they can't prevent it completely, and they can concentrate at the target, at the tumor. Either passively because of their size, that's what I showed you before, or you can actually decorate them with affinity ligands, so chemical zip codes that will make them enrich even further. So this is, as I said, this should increase the concentration at the site of the tumor by about 10 to 100-fold and reduce side effects systemically. So this is a very active field, and one that we've also participated in, and we're super excited about. I should say, as a caveat, as a field, all of us who are doing this work still have limitations. And by that I mean, of the injected dose that we give, sort of typical concentration at the site of the tumor is 1% of the injected dose. Now, this is still 10 to 100 times better than the free drug, but it's still only 1%, so that means 99% of our targeted cancer nanotherapy is not going to the tumor. So in our group, we got very interested in what could we do to further amplify deposition of this chemotherapeutic at the site of disease. And for this, we sort of started thinking about what else is cool about the nanoscale. So what else is cool about the nanoscale is materials completely change their properties, and you can try and use that to create systems of nanomaterials that can do something like amplify deposition. So let me walk you through what that looks like. This is a movie. This is a block of gold. Looks golden at the macro scale. If I shrink this material down to 100 nanometers, it turns optically red. If I change the shape of it into a rod-shaped material, it becomes an antenna for light. And the shape of this material, whether it's long or short, dictates what wavelength of light it's sensitive to. So this is a near-infrared sensitive one. This shorter, fatter one is a green light, visible light sensitive one. So this happens, for the physicists in the crowd, this happens because the electrons are really mobile on the surface of this material, and the shape of the material dictates the resonant frequency at which they get excited. Okay, so now I have a material that I can activate from the outside world and maybe put in a system to increase deposition of a targeted chemotherapy. The way we imagine this working is just the way the body amplifies deposition of materials in tissues. This is a great example of the way nature actually uses communication in your tissues to amplify deposition by recruitment of white blood cells. So what happens here typically in an infection is you can have a macrophage in a tissue, it's ingested a microbe, and it starts giving off chemical signals through the tissue, through the endothelial cells that line the blood vessels, and those endothelial cells get activated. They change their surface expression, and they can now recruit circulating cells like neutrophils to the site of disease where they amplify their deposition. Right? So you're sending a signal, and you're listening for a signal, and these two populations are communicating through chemical signals. So we started thinking about, can we make nanomaterials that are highly specialized, that communicate through the body through chemical signals? And one candidate are those nano antennas that I told you about. They're very efficient transducers of energy. You don't need very much of them to start a chemical cascade. So this is how we sort of play this out in the nanomaterial space. You have a signaling nanoparticle that's sending the signal through the tissue. It's found the tumor. And then a drug-loaded particle, and it's just listening for the signal. It's the receiving nanoparticle. Okay, and these two things act together instead of acting alone. And when we do this in a mouse model with a cytotoxic called doxorubicin, which is a well-known chemotherapeutic drug, and we use the gold nanoantenna and a liposome loaded with this doxorubicin, we get, for every one little nanorod, we get 35,000 molecules of drug at the site of tumor through this communication. If you do this in a mouse model of breast cancer, you can see in this scenario that you can treat these tumors very effectively. And if you have the same amount of drug in the system, but the particles aren't communicating, you get virtually no effect. Right? So this amplification actually drives the lion's share of the therapeutic effect. Okay, so we started doing this, and the students got very excited because we started thinking about systems of nanomaterials. Right? Each injection is actually 10 to the 13th nanomaterials. So here I have two different flavors that are interacting, but you can start to imagine what can we do in a system where we have many, many different kinds of nanomaterials. About this time, a postdoc in my lab came to me from Switzerland, and she is actually a swarm engineer. She works on flying robots and trying to get them to swarm. And her inspiration was flocking birds. This is showing not on the screen. Okay, there are birds flocking. this image, but imagine any kind of swarming behavior. Hives, bees, birds, termites, ants foraging for food. These are all emergent behaviors that come out of a collective. And the way this works is that you don't need a leader. You don't need positional information. All that you need to endow these agents with is information about how they should respond to their neighbor. And this got us very excited about what we can do in nanomaterials. And so now we've been making nanomaterials that assemble, disassemble, communicate, and have collective properties. So this is sort of very early work that's happening at MIT. The computer has finally caught up with us. There are the birds. And then this is a collaboration with a colleague of mine who's a roboticist at Harvard, where she's made robotic, simple agents where we give them very little information. We only tell them how to interact with each other. Okay, and so you can see that we can get them to perform complex programs. So in this next program, what you're going to see is a follow the leader program. They have no positional information. All they need to know is to follow the one closest to them and stay a little bit ahead of the one behind them. And you can get these complex behaviors. And these are the kinds of things, these very simple functions that we can actually program into the nanomaterials themselves. This is a program for dispersal. And the program is very simple. Go as far away from your neighbor as you can. And that ends up being the collective function. This is a program for synchronization. Okay, so this is a program in our group that we work on. We call this swarming nanomedicine. It's actually very difficult to develop intuition around this. So we thought about mining the crowd, and we've turned this actually into a game. There's a game, it's an app, you can download it, it's called nanodoc.org, and on the back end of the game is a computer simulation that carries all the nanomaterial physics and all the transport details and the tumor biology that I told you about. But as a gamer, you don't need to know that. All you have to do is play the game and try and get as many nanomaterials as you can into the tumor, and then we, on the back end, we mine that for inspiration to design new nanomaterial functions. Okay, so this is all very exciting, but I had another student a couple years ago named Tal Danino, who's now a professor at Columbia, and he came to me from a synthetic biology lab. And he said, you know, the problem with your strategy is that you will never get more than you put in, right? And the cool thing about nature, if you're really inspired about nature, is that biology can grow. What if you deliver something where you deliver one of them, and then you will get more than one of them? And so for that, we turned to synthetic biology. So here what we've done is actually design bacteria that make chemotherapeutics inside the tumor. And the way we do that is we take this strain. It's commercially available over the counter in many different forms. It's an old strain of E. coli that was actually obtained from a soldier in World War II who was resistant to GI disorders. It's been used extensively to treat IBD, and we engineered this organism to make chemotherapeutics on site. And we wanted to do this in a very specific way. The way we wanted to do this is we didn't actually want chemotherapeutics to be released everywhere the bacteria went. We only wanted chemotherapeutics to be released where the bacteria had enough nutrients and immunoprivilege to grow. And it turns out that they can grow inside tumors. It's low pH, it's low oxygen, it's nutrient rich, and they can hide from the immune system. And this is an emerging field of the so-called tumor microbiome. And we wanted to take advantage of that. So if we knew if they could grow in tumors, that we wanted them only to turn on their chemotherapeutic program there. And so in order to do that, we took advantage of a bacterial program that some bacteria already have called quorum sensing. And this is where they make chemicals and they can detect how many there are of their neighbors. And they can drive gene expression. So what we do is we have them quorum sense. This is the circuit. And when they grow up to a critical mass inside the tumor, they lyse themselves and they dump their contents. Okay, so it's a quorum lysis program. And just to sort of give you a visual, this is actually what it looks like. On the left, these are these synthetically engineered bacteria in a microfluidic device. And when they detect a quorum, they first turn green and then they lyse. And then the few stragglers grow up again, and they turn green, and then they lyse. And that's them detecting that they're in a quorum. And this is very precise. You can control the periodicity of this. You can actually make a bacterial oscillator. And you can see that here. So if we do the periodicity of this, and we can tune the period of this depending on what we think will be most therapeutically efficacious. So we test this in the lab, and you can see here that they very effectively kill cancer cells in a microfluidic device. We actually, since we were doing proof of principle and we really wanted this to work, we put in 3 toxins at once. We put a pore-forming toxin in, an apoptosis trigger, and a chemokine that would recruit the immune system. So 3 things that get delivered upon quorum lysis in this material. And then we put them in a mouse model of liver metastasis. This mouse model of liver metastasis actually glows. So you can see in this middle panel here that those are the tumors. And when we give these bacteria orally, they get into the gut, they spill over into the gut during a feeding, and they grow up. They virtually overlap with tumor location. So you can do 2 things with that, actually. You can make a sensor. We've also made a sensor. Or you can make the therapeutic that I just told you about. If you look elsewhere in the body to sort of test this idea that the tumor is an immunoprivileged site where they're growing, we grind up all the organs in this animal, and we can't find bacteria anywhere else. They are only in the tumors. And we have a therapeutic survival result in these animals, where the bacteria, together with chemotherapy, can allow a survival extension. So I'm not saying that this is ready for patients, but it's a very interesting, intriguing idea for how one might make oral probiotics that could be next-generation tumor detectors and therapeutics. So the last thing I just want to touch on is that we're not alone in doing this work at MIT. This summer we started a center for cancer nanomedicine, and this is a collection of our faculty. We're working on a lot of different interesting angles on this problem: cancer vaccines, cancer immunotherapy, intraoperative imaging using next-gen materials and instruments. So I welcome you to come to our website and learn more, or come visit. And just to sort of wrap up, the future, I think, of miniaturization in medicine is very promising. And in particular, I think that's the case because I've just shown you a snippet of this length scale. I showed you nanomaterials that were sort of 10 to 100 nanometers in size, but it turns out in engineering, there are a whole world of miniaturization tools that are pouring out of manufacturing and material science and electrical engineering. And you hear about some of them, and you'll hear about 3D printing, you'll hear about microfluidics, but if you sort of collect them on this length scale, you realize this is an incredibly rich and fertile ground on which to innovate cancer diagnosis and therapy approaches. And really, if you think about disease tissues, this is the length scale where the action is. A single receptor is 10 nanometers, a single cell is 10 microns, collectives of cells are 100 microns. So these tools together are allowing us to make tissues, to fabricate them, to model human diseases in unprecedented ways, and to monitor them by making materials small enough to actually go into the disease site and make a measurement that we couldn't make before. So thank you for your attention.